a nucleic acid aptamer is obviously security of supply. So for industry, this is a really big problem. You don't want to be in a situation where you've been working with a product that's an antibody base, for example, and you have a hybridoma line that works absolutely fantastic until your freezer dies, and then that hybridoma is dead, or over a couple of years it starts to change through repeated culture, and what you find is that what the product you're selling now is not the same product you were selling 10 years ago. And as Ed pointed out, these aptamers are actually just strings of information, so you can actually just store that as a Word file. There's nothing physical you need to store. So as we say, we can email that to sites all around the world. There's nothing to distribute. You can just send that information to the customer, and they can archive it and just shelve it until they're ready for it, so you know that the product you make in 20 years' time is going to be exactly the same product that you're making tomorrow. So that's a really important point for these things. They're also very robust in terms of their stability. So chemical synthesis of aptamers is a really well-characterized process now. So the strict quality control that can be put around that assembly process is phenomenal and is much better than most other technologies. I don't have time to go into all of the benefits here, but if you've got any questions about this, again, come and see us on our stand. But these are really helping aptamers to find their way into almost every area of the life sciences, and people are actually starting to see aptamer products out there without even realizing it. So there's examples of that out there that I'm happy to discuss later. As Ed mentioned, in the biomarker discovery process, you're really playing spot the difference, and it's all about helping you to spot that difference. So rather than looking for a needle in a haystack, let's try and use a magnet to find that needle. So as Ed pointed out, a more traditional approach these days is to look at mass spec, but what you could do with that sort of processing is really throw out the baby with the bathwater. So if you go through all this processing to get to a usable mass spec, you might have thrown away what you're actually interested in. So what we do is say, well, we don't need to do any of that processing. Let's just go in with the raw material. So in this case, looking at healthy versus diseased cells, and I'm going to expand on the properties of that in a second. So this is the same animation that Ed showed you before about how we go about getting disease-specific aptamers. And again, to show you the same sort of data to say this process actually works. You know, we can take this sample, this can be a tissue. We've done this with tissues, we've done it with cells, we've done it with organisms. It doesn't really matter to us. But where we can expand on this is, in that simplistic view, you've got one cell with one biomarker, and that's not really realistic at all. You're much more likely to have something like this where you've got multiple markers. But that's actually a benefit, because what we will have is multiple aptamers to multiple markers on that cell line. So you could see here, you could start to get an idea of profiling. So you could say that this patient, for example, has 2 yellows, 2 greens, and a red. Fantastic. Well, here's a different patient, and everyone will know that one patient is not the same as another. So this patient's only got 2 yellows and 1 green and no red. So that would then say, well, actually, the better biomarker for this disease is the yellow. So let's target that. But in our studies of patients, we can say, well, this patient's got these level of markers, this one's got these level of markers. So you get 2 tools out of the same product. But we can go a step further than that. When it comes to target validation, you could say, well, we want a drug that targets the purple protein for the yellow aptamer, so let's use it to study that. Well, and you can see when we treat the cells, we can now use the aptamers for target validation purposes as well. We can say, yes, when we target that purple protein, the yellow aptamer has gone down. So we've essentially got multiple products out of one process. We've got a biomarker discovery, we've got patient profiling, we've got patient stratification, and we've got target validation, all out of the same process. And again, I don't think that this is a technology process that can be boasted by the other technologies. And here's some examples of that working. So hopefully, maybe you can make out there, but basically we've got a nice 2D page here stained with our various aptamers, showing some proteins go up, some proteins go down, some stay the same. So patient profiling is realistically possible. Something else that we're working on at the moment that we don't think we can do with any other technology is in-cell real-time tracking of cellular proteins, metabolites, turnover of small molecules, all that sort of thing, in a simple assay format that says if the cells are producing protein X, they start to fluoresce. As soon as they stop producing it, they stop fluorescing. So this gives you a real opportunity to monitor exactly how your cells are responding to a particular stimulus. So that could be disease or treatment or whatever. So Ed's got these posters up. They're also on our stand, so anybody who has any questions about this or would like me to expand on any of this technology, please come and find us, and we're happy to talk about it further. And I hope that was 5 minutes.
Thank you.